Selamat siang, Bapak Ibu dokter sekalian, peserta panitia. Izinkan kami kembali untuk membawakan sesi berikutnya, yaitu pemaparan dari prinsipal kita, salah satu dari prinsipal kita, yaitu dari YHLO, yang akan langsung dibawakan oleh manajer marketing untuk wilayah area Asia Tenggara, yang akan dibawakan oleh Mr. Samuel Yu. Ya, beliau sendiri merupakan lulusan magister untuk jurusan Biological Science dari Institute of Technology Illinois di Amerika Serikat. Beberapa tahun beliau habiskan di bidang in vitro diagnostik. Kemudian sekarang berposisi sebagai Clinical Manager Marketing di YHLO. Iya. Barangkali langsung saja tanpa memperpanjang lagi, kita langsung dengarkan bersama-sama pemaparan untuk alat iFlash 1800 maupun UNICELL-S yang akan langsung dibawakan oleh Mr. Samuel Yu. Mr. Samuel Yu, the time is yours.
Okay. Okay, thank you. Thank you, Setia, for the invitation, and thank you all for coming today's conference. So my name is Samuel Yu. So I will present the principle of the neutralization antibody from YHLO. Okay, so before that, I will give you a brief introduction about myself. I'm a master in the biological science and also achieved the project management professionals. I graduated from the US Illinois Tech. I have 10 years working experience in the IVT field and also the former product manager in the similar company. And right now I work as a clinical marketing manager in YHLO. Okay, so before my presentation, I want to share some data. So this is the data from Johns Hopkins, and the data updated to March 24th. So the global, actually, the situation is very severe right now. More than 124 million people affected by the coronavirus. And some countries are very dangerous right now. For example, the US, Brazil, some countries with large population. Okay, so the situation is very severe right now. So today I'm gonna talk about the solution to the vaccination, detecting the vaccination, the neutralization antibody. Okay, so my slide will divide into 3 parts. The first one, I will introduce the neutralization antibody from YHLO. And the second, I will share some common method that used right now for the detection. And the third one is the comparison among YHLO method compared to other common method in the market. Okay, so the structure of the SARS-CoV-2, actually we are very familiar with that. Inside there's and all reagents okay, cell and reproduce itself and infect people. So the entry of the virus is very important. If there's a way to prevent the entry of the virus, and that will be very helpful to defeat this virus. So talking about the viral entry, there are two major parts we need to focus. Is the binding receptor on the host cell, we call it ACE2, okay? And the surface from the surface of the virus, S protein, we call it RBD domain, okay? So this binding between RBD domain and ACE2 make the virus come into the cell. Okay, so if there's some antibody against the binding between RBD and ACE2, that will prevent the cell come into the cell. Okay, so that's why we are introducing the detection of neutralization antibody, because the neutralization antibody, the function is to prevent the binding of ACE2 and the S protein. Okay, there are a lot of vaccines right now on the market. Some of them are inactivated vaccines. Some of them are RNA vaccine. So all the vaccines actually work at a similar principle. So they will trigger the body to produce large amount of antibodies against the virus. Okay, so large amount of the antibodies consist a lot of different types of antibodies, including neutralization antibody we talk about, and also other antibodies that have no neutralization functions. Okay, so like I said, after the vaccination, there are a lot of antibodies generated, but some of them they target against the spike protein, S protein, S1, S2, RBD, and some antibodies may against other parts of the virus. But which part of the antibody is sufficient for the in the protection? So we suggest that the S protein, especially the neutralization antibody, makes an important role in the prevention. Okay, so not all antibodies against the S protein that are induced by the vaccines are neutralizing antibody, but only neutralization antibody makes an important role in the vaccination. Okay, so according to the research, I found some research papers. Right now, there are a lot of antibodies that were generated after the vaccination. So the research said they collected more than 100 antibodies that after the vaccination or after the infection. But the researcher found that only 11 from them have neutralization functions. So that means not all the antibody generated from the vaccination can work as the neutralization antibody. Okay. So that makes the differentiation and the quantification of the neutralization antibody very important if we want to monitor the effectiveness of the vaccination. Okay, so why should we detect the SARS-CoV-2? So actually we have three main clinical values. The first one is to evaluate the COVID-19 vaccines. Okay, evaluate its efficiency or evaluate the protective effect after the vaccination. Okay. The second is about the analysis of a herd immunity status, because we know that right now some countries, they have a large population affected by the virus. So they want to cope with the virus through the herd immunity. So the analysis of the neutralization antibody can know exactly the big data of the population. Okay. And third one is that to monitor during the convalescent plasma treatment, because we know that some convalescent patients may produce large amount of antibodies. So through their plasma treatment, we can still we can offer a very good treatment to the clinic, okay, and to evaluate the treatment by using the detection of neutralization antibodies. Okay, so second, I will talk about the method for the detection of neutralization antibody right now. So first one is gold standard, the PRNT, okay, the plaque reduction neutralization test, which is right now used in the laboratory actually. So the detection PRNT is it will use the serum with patient sample with the neutralization antibody, and it will put into the virus, and after the culture, it will place on the plate. And by counting the plaque on the plate, we know what's the effectiveness of this neutralization antibody. But the limitation of this, though this is a gold standard, but this limitation is obvious. It takes long hour, and also it's a manual operation. It's not standard. It has no standardization procedures right now. Okay. So right now we YHLO we can introduce a new method. Later I will talk about, which has a higher sensitivity and also have very high specificity. Okay, and also avoid very cumbersome operation and save the time for the operator, also save a large cost for the lab. Okay, so that's why today I'm introducing the iFlash system from YHLO. It's a clear system. The result can be very comparable to the PRNT result and also some ELISA result in the market. The antibody is purified and can be very specific and also can be very sensitive. Okay, so the detection principle for the iFlash system, actually the reaction takes place in the liquid system. Okay, it's a complete liquid system. Unlike ELISA, because ELISA is coated on the solid surface, but the iFlash, the clear system, reaction takes place in the liquid system. It used the magnetic microbeads as a separation tool. So the microbead on the surface, the antibody antigen were coated on the surface. And also the signal is triggered by the acronym ester, which is a very sensitive light signal. So with magnetic microbeads and the acronym ester, we can create a very sensitive system to detect neutralization antibody. Okay, so the detection principle is based on a competitive method. Okay, so as I said, the microbead is coated with the antibody with actually the RBD. Okay, and the acronym ester is coated with ACE2. So normally RBD will bind to the ACE2, right? And the function of neutralization antibody is to prevent the binding of RBD and ACE2. Okay, so that makes the neutralization antibody will complete with ACE2. Okay, so the principle is designed as a competitive method here in the iFlash system. Okay, the iFlash NAB we have tested in a lot of hospitals in China and also internally in YHLO. So we have data suggested that the agreement with the PCR positive patient or patient after vaccination, the agreement is as high as more than 90%. And also the specificity is as high as 99%. Okay, our product right now used worldwide. We have many customers from Europe, some from Asia Pacific, from Middle East, from America. They are using our product. This is a hospital, actually it's a reference hospital in Europe. called Italy Sacco Hospital. So they have done the evaluation with our neutralization antibody against the PRNT method. Okay, so they have published this paper, have a conclusion that YHLO's NAB test have a very good correlation with PRNT. Okay. Beside the iFlash system, we also have a flexible and convenient smaller system. We call it UNICELL. So UNICELL is designed for convenient use of the detection. So they provide a convenient operation, provide a flexible, accessible ability to the lab, and also provide efficient detection time. They can finish the test with 15 minutes. Okay, so both iFlash and UNICELL can offer a comprehensive solution for our clinical workers. Okay, so the UNICELL is a little different from the iFlash system. It uses a strip to detect the NAB. So the sample will add into the strip, and it will detect using the solid surface. Okay, but the principle from iFlash and also the principle from the UNICELL, they are the similar. They all use the competitive method. So the NAB in the sample will compete with the ACE2 and the binding with ACE2 and the RBD. Okay, so also we have done a lot of experiment, and the agreement with the patient after the PCR test, PCR positive patient, or with the vaccination patient, the agreement is about 90%. Okay, and also compared to the PRNT, we have more than 90% same result. Okay, the last one, I want to introduce some method in the market. Like I said, there are some other method. For example, there are antibody against SARS-CoV-2 RBD, like Roche, like a ELISA method called GenScript. They also have a NAB test. So what is the advantage from YHLO neutralization antibody? Okay, so first I want to compare to the reagent that against the SARS-CoV-2 RBD. Okay, so first of all, I want to emphasize a concept that a lot of tests is against the SARS-CoV-2. So for example, the IgG, SARS-CoV-2 IgG, SARS-CoV-2 IgM. So we also have this kind of test. Okay. And another one is called antibody against the S protein, or the antibody against S protein RBD. Okay, for example, Roche, they have the similar test. Okay, and the third one is about antibody against the neutralization antibodies. Okay, this kind of antibodies like GenScript, ELISA method, like YHLO, iFlash, NAB. So we have this neutralization antibodies. So these three tests, they have a different targeting. Okay. So with the understand of different principle, we know that Roche used the method. Okay, they are detecting the antibody against the S protein. Okay, so actually Roche detecting not only the antibody against the neutralization antibodies, but also other kind of antibodies, like they don't have neutralization functions. Okay, so this is the Roche. Okay, they detect all the antibodies against S protein. Okay, but YHLO use a competitive method. So YHLO only detect the neutralization antibody. Okay, only detect the neutralization antibody. This is the difference. So if we have YHLO test, we can detect antibodies, neutralization antibodies Okay, and neutralization antibodies will prevent the cell from the entering the virus from the entering of the cell. Okay, but like Roche, not all the S protein, not all the antibody against S protein will prevent the virus from entering the cell. Okay, only some of them can do the function. So not all antibodies that bind to the RBD are neutralization antibodies. Okay, so this is the limitation from SARS-CoV-2 S reagent. Okay. There's a paper published in the Nature. Okay, it says that after the infection of the virus or after the vaccination, especially the patient after 6 months, the neutralization antibody will decrease generally. Okay, so you will see that this is the data. So patient after 6 months compared to the patient after 1 month, their neutralization antibody will decrease dramatically. Okay, they will decrease dramatically. Also, this paper published in the Lancet, they found a similar situation that the neutralization antibody was significantly lower after 6 months. Okay, so this is common in the after the vaccination or after the infection. So we have done some data, some experiment in Europe, and from the data left, we see that after 6 months, the patient with YHLO NAB detection, they have increased. Okay, they increased very dramatically. They increased dramatically. Okay, but with the Roche antibody against S cell, they remain the same. They remain the same. So that's why Roche is not detecting the real neutralization antibody. They are detecting all the antibodies against. So it's important. Actually, effectiveness. Okay, so another company, the previous speaker, they also used the gene script detection. So gene script, they use a ELISA method, and their principle is similar with YHLO. So, so from the principle, they are detecting the same antibody group with my limitation from gene script is with their method, ELISA. So basically, advantage in the higher sensitivity, and also the microbeads is mimic the same situation in the human body, while the gene script ELISA use a plate and all the antibody, all the material are coated on the surface. Okay, so this is the advantage over the ELISA. And other advantages like the microbeads, like I said, is free to move, small like the reaction in the body. Okay, and also the linearity have a wider range compared to the ELISA. Okay, and with the automation, CLIA used a complete automation system, while ELISA still require a lot of manual work and take a longer time compared to the CLIA system. Okay, so also we have data from Europe. We have done some comparison with gene script. So we'll still see that the YHLO, so after 6 months, the neutralization antibody have a dramatic decrease. But with gene script, we also see it's not dramatically, okay? That can also prove it's not detecting the real NAB, while the gene script is detecting the NAB, but it's not sensitive compared to the CLIA. Okay, so as a Chinese company, we are the first to stand out to fight against the COVID-19. We have start the research in the February 2020, and still we send our product to a lot of countries in Europe, in the Middle East, to help them to cope with the pandemic. We still cooperate with the military from China and to help them evaluate the vaccine development. For example, the vaccine developed by the Sinovac, they are using our iFlash system to do the evaluation. Okay, so right now we have a lot of reference labs that are using the YHLO NAB. For example, the Karolinska University Hospital from Sweden, a Limbach group from Germany, and a lot of hospitals there in the world are famous. They're using the YHLO NAB to do the evaluation. Okay, so we have Italy, Germany, even Japan. We have a lot of reference lab. They are proved. They have proved the quality from YHLO. Okay. So that's the today's presentation, and that's the information from YHLO. And thank you for your kindly attendance. Thank you all. So any questions?
Yeah, thank you very much, Mr. Samuel Yu, for the elaboration for the presentation. Ya,
Thank
mungkin
you.
para hadirin ada yang ingin ditanyakan atau peserta yang mengikuti secara online boleh langsung drop pertanyaannya di kolom chat. Barangkali dokter ada yang ingin ditanyakan? Silakan. Ya, jadi tadi sedikit pemaparannya kalau boleh kita simpulkan terdiri dari 3 bagian, introduction, yaitu penjelasan singkat mengenai proses infeksi COVID-19 tadi maupun cara kerja dari tubuh memproduksi antibodi. Kemudian yang kedua yaitu penelitian lebih lanjut di mana-mana saja alat untuk NAB ini diteliti. Salah satunya tadi di Italy International Hospital di Sacco, yaitu tepatnya di kota Milan. Beberapa perbandingan dengan alat yang serupa juga telah dilaksanakan, yaitu dengan ROS tadi. Di situ juga diteliti dengan gold standarnya, yaitu PRNT. Kemudian di bagian terakhir beliau memaparkan tentang comparisonnya dengan alat-alat yang sekelas. Tadi ada GenScript, juga ada ROS.
Baik, ini ada satu pertanyaan, tapi dengan bahasa Indonesia nanti moderator translate ya.
Baik, terima kasih. Saya mau bertanya, kalau tadi Dr. Dewi menyatakan untuk Mindray dan Roche sudah ada satu nilai konversi dari AU ke nilai yang dinyatakan oleh WHO sebagai BAU itu, apakah untuk iFlash 1800 ini sudah ada penelitian untuk mengarahkan nilai standar tersebut? Berapa pengalinya? Terima kasih.
Ya, mohon diulang dokter yang terakhir tadi untuk yang di gimana?
Ya, untuk pemeriksaan metode kita di iFlash 1800 ini, kan kita masih menggunakan satuannya masih AU per mililiter. Kalau dari WHO sudah dinyatakan bahwa untuk Mindray dan Roche sudah ada nilai pengalinya untuk menjadi BAU sebagai suatu satuan unit secara internasional. Nah, apakah sampai saat ini dari prinsipal sudah ada penelitian yang menunjukkan berapa faktor pengali tersebut? Terima kasih.
Tadi yang dari WHO bau tadi dok ya?
Iya, baunya itu
nilai
tadi.
Baunya tadi dok?
Iya.
Oke. Ya, Mr. Samuel Yu, is my voice audible? Yeah, there is a question from our participant. in a previous speaker, Dr. Dewi, she informed us that WHO has released the conversion number from AU per ml, antibody unit per ml, that is converts to smell number. So in our iFlash 1800, is there any similar conversion with AU per ml converts to smell number? Thank you.
Okay, thank you. So I want to confirm. You want to know the comparison with whom? From iFlash 1800. So the comparison with which one?
Yeah, can you please pause it?
Yeah. So could you repeat the question? The comparison with which one?
Comparison from the instrument. The instrument released the AU per ml number, correct? Our instrument released the number of NAB in form of AU per ml. AU per ml, antibody unit per ml.
Okay, I understand. So you mean the unit from other company like UML, and from iFlash is AUML, right?
Yeah. iFlash released the AU per ml number.
Yes.
As we have already know from previous speaker, she said that she informed that WHO has released the coefficient number that is converts from AU per ml to smell number.
Okay.
Do we have
Okay,
the same
I understand.
method, by the way?
I understand the question. Okay, so so right now the unit from YHLO is AUML. Okay, just to be confirmed. But we will still need the calibration material, calibrators, international calibrators to do the linearity evaluation right now. Okay, so after the evaluation of the linearity range, so we can claim our UML. Okay.
Ya, terima kasih.
Ya, thank you very much. Jadi tadi jawabannya masih kita coba cari untuk nilai koefisiennya dari alat ini karena belum ada untuk metode, untuk parameter NAB ini. Jadi AU per ml masih hanya diterapkan untuk di SRPD. Begitu. Terima kasih. Mungkin ada pertanyaan berikutnya.
Yang online, Pak, yang online.
Yeah, next question, Mr. Samuel Yu. There is a question in the chat box which asks, what is the cut-off value of our YHLO instrument?
Sorry, could you repeat the question, please?
Yeah. What is the cut-off from the NAB parameter at our instrument?
Okay. So right now, the cutoff of the YHLO iFlash system is 10 AU/ml. Okay. The 10 AU/ml is defined with a large sample evaluation with a group of healthy people and a large group of people who are infected with the virus, and also people after the vaccination. Okay, so 10 AU ml is the cutoff, but we suggested that 8 to 12 as a gray zone, from the test.
Ya, jadi nilai untuk cut off alat ini adalah 10 EU per ml. Jadi di atas itu masuk pada gray zone atau dikatakan sudah meningkat, dengan sebaliknya di bawahnya juga demikian. Ya, mungkin ada pertanyaan dari dokter untuk peserta offline.
Mas Restu, saya mau bertanya.
Ya, dokter.
Apa hal-hal yang mempengaruhi proses pembacaan alat kita? Apakah misalnya kalau biasa kan lipemik, hemolisis, atau ada faktor-faktor lain yang mempengaruhi proses pembacaan pada alat kita, sehingga kenapa hasilnya bahkan kurang dari cut off gitu? Karena beberapa sejawat yang periksa NAB pada hari ini itu ada yang kurang dari cut off. Apakah ada yang mempengaruhinya gitu, selain lisis ataupun lipemik? Terima kasih.
Faktor lisisnya tadi kan?
Ya, apa ada faktor selain hemolisis atau lipemik gitu yang mempengaruhi? Apakah karena obat atau karena apa? Kenapa didapat kadarnya kurang dari cut off? Gitu. Terima kasih.
Yeah, next question, Mr. Yu. Is there any external factor that can interrupt the reading method of this instrument? For example, we previously test the NAB from our participant. Is there any external factor, for example, consuming the medicine or any external factors? Thank you.
Okay. So right now we don't have such experience in the factor, but I guess the people after the vaccination can do the evaluation. So if this more data is coming, we can know more about this affection.
Yeah, because previously some of our participants has tested the NAB from our UNICELL-S, and the number that is released from the instrument is below the cut-off value.
Because the UNICELL and the iFlash, they did the evaluation separately, So, but UNICELL and the iFlash, they are using the same material for the R&D, the research and development. They use the same material. So basically, the performance of iFlash and the UNICELL, they are similar. They are similar to each other, but the methods are different. Right? The methods are different, so they have a slightly difference because of the different platform. Yeah.
Ya, itu tadi, dokter. Mungkin faktor eksternalnya, kalau dari penelitiannya mereka berdasarkan hasil research and development-nya, baik alat kecil yang kita gunakan di sini maupun dengan alat besar, hasil research and development-nya sama, tapi tetap ada faktor-faktor eksternal yang menentukan, dan ini masih dalam proses pengembangan mereka sampai dengan saat ini. Barangkali itu. Ya, baik. Barangkali kita cukupkan saja untuk sesi tanya jawab ini tadi. Mr. Samuel Yu, we do appreciate for your elaboration of your presentation. We thank you very much. We hope that we can meet in other occasions. Thank you very much.
Thank you. Thank you all.
kita akhiri saja untuk simposium kita pada hari ini. Saya Setia Anugrah Pratomo, terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.